Hi everyone. This video is a comprehensive interpretation of research paper published in month of April 2016 in Journal of Proteomics. The title of a paper is Proteomic Analysis of Human Tooth Pulp Proteomes, Comparison of Caries Resistant and Caries Susceptible Person. We hope you all enjoy our presentation and learn something new about proteins.
Kunal, how are you doing in your proteomic report?
I'm doing well. How about you?
Well as well.
Good.
Do you want some chips?
Yeah, why not? But I think after eating this one, I should go and brush my teeth.
Come on, brush your teeth, but you only brush your teeth twice a day.
No, I started brushing after every meal. I read an article last week about proteomic analysis of human tooth pulp, and it's very informative.
Yeah, the one about the comparison between caries resistant and caries susceptible person.
Yeah, it's very good.
Oh yes, why don't we analyze it?
Yeah, just let's do that.
Dental caries are a worldwide problem. Research has shown that over 90% of adults experience dental caries. Caries continue through adulthood, with 90% of adults experiencing caries on enamel and root surface.
Dental caries is a multifactorial disease that can include the participation of cariogenic bacteria, nutrients present in the dietary sources, and salivary components such as enzymes and proteins. American studies have revealed that having a low-quality diet, reduced intake of vitamins and minerals, the physical activity, and smoking status are associated with the teeth loss. As I have said, this data reveal that only 10% of population is protected against dental caries. This anti-caries resistance could be caused by different factors such as the immune system, plaque composition, specific dental care, among others. But not the least, resistance that could be caused by specific protein antibacterial agents inside the tooth tissues. Saliva is one of the major factors responsible for the prevalence of dental caries. It affects the colonization and growth of bacteria. Saliva covers the oral hard and soft tissue. It includes lactoferrin, lactoperoxidase, lysozyme, and antimicrobial peptides. Therefore, saliva protects overgrowth of few pathogens. There are few studies that focus on the saliva composition and bacterial growth. However, Vitorino and collaborators, as well as young teamwork, agree that cytidines, amylase, immunoglobulin A, lactoferrin, and acid proline-rich proteins are positively correlated with caries resistance. The proteome of human dental pulp has been extensively investigated since 2008. In 2005, Pakonen and collaborators studied the first proteomic human dental pulp, locating 96 proteins using two-dimensional gel electrophoresis followed by MS-MS. The aim of the present study was to use DISH comparing teeth samples between caries-resistant and caries-susceptible group of people. This will enable the investigation of potential candidate proteins responsible for the resistance of the tooth dental care in humans.
For sample preparation, permanent human third molar with closed apexes of 12 adult aged from 24 to 40 years were extracted for various clinical reasons. The obtained teeth were then divided into two groups depending upon DMFT score, which is decay missing fill index. In one group, teeth from 6 adults, 3 males and 3 females with a DMFT score of 0 were collected and categorized as caries resistant R group. These people never had any tooth caries in their mouth. On the other hand, teeth from 6 adults, 4 females and 2 males with a DMFT index ranging from 4 to 15 were collected and categorized as caries susceptible S group. These patients were cured for the tooth caries presence in the past. After a series of procedures, the dental pulp were removed and from all 6 teeth and pooled. For 2D Dij experiments, the pulp sample was homogenized, mixed with lysis buffer, thiourea, protease inhibitor, and finally filtered, and the protein concentration adjusted to 1 mg per ml with a pH to 8.5. 160 microliter of protein sample from caries resistant and caries susceptible groups was labeled with Psi5 and Psi3 dye respectively. Psi2 dye was used to label an equal mixture of protein sample 80 microliter from caries resistant group and 80 microliter from caries susceptible group to form an internal standard. Then the labeled samples were combined in the ratio of 1:1:1 and subjected to 2 large and 2 narrow IPG strips. 3 different gel images were obtained from one gel at appropriate wavelength by using a Faros FX scanner. and then analyzed by PDQuest software. Spots with differential expression were excised and subjected to mass spectrometry for protein identification. The expression changes in identified proteins were later confirmed by Western blotting.
They used two formats of IPG strips. This allowed the detection of many spots. First, they used large format yields with a broad range between 3 to 10 pH. Secondly, they used a narrow range between 4 to 7 pH. The samples, R and S, were labeled with Psi3 or Psi5 and were run in both 2D dye yields. The average number of spots detected on the large yield was 360 on the smaller one. Protein spots exhibited migration positions from 15 to 250 kilodaltons. However, as we can see, they are clustered between 20 and 150 kilodaltons. In the large yield, 9 protein spots exhibited significant differences when comparing the carrier-resistant and carrier-susceptible samples. Out of 9 spots, 5 exhibited greater expression, marked in a solid line, and 4 show lower expression, marked with a dashed line. Mini-yield image analysis shows 7 protein spots with significant differences. Out of 7 spots, 6 show greater expression and only 1 spot exhibited lower expression of carrier-resistant sample compared to carrier-susceptible sample. The spots appearing either in the large format or in mini format yields in the range of 4 to 7 pH and 30 to 80 kilodaltons regions were further identified by mass spectrometry. Only protein spots displaying significant changes, p less or equal to 0.05, were considered. Spectra were reached against the IPI human database using the mascot engine. As you can see in this table, we can identify the spot number, accession number, gene name, protein name, mascot score, theoretical molecular weight, and pi values. The identified proteins were categorized according to their biological processes functions: protein metabolism, energetic metabolism, cell growth, cell communication, and transport. As I have previously mentioned about greater and lower expression, we can see that in spot number 1 corresponding to alpha-1 antitrypsin was upregulated by a factor approximately of 6.2. Similarly, another spot of alpha-1 antitrypsin was upregulated by a factor of 4.3. However, some spots display downregulation. For example, spot number 6 corresponding to serotransferrin was downregulated by a factor of 0.43. In some cases, MS analysis revealed the same identity for multiple protein spots appearing as separated entities into the E-gels. This indicates the possibility of the presence of various isoforms of these proteins. It could be due to post-translational modification. The majority of the isoforms of any particular protein exhibited an identical expression, either up or down regulation in the carrier-resistant group. For example, annexin, A2, serotransferrin, vimentin, with the exception of alpha-1 antitrypsin, were upregulation as well as downregulation spot observed. After 2D dye, they performed Western blot of alpha-1 antitrypsin content in sample obtained from carrier-resistant group, shown in the left side, or carrier-susceptible one, shown in the right side. The number spot represents statistically significant up or down regulation. As we can see, the expression pattern of alpha-1 antitrypsin spots were consistent with the 2D dye results.
In this study, soluble proteomes of carrier-resistant and carrier-susceptible tooth pulp samples were separated with 2D dye. 12 proteins were found to be differentially expressed between the carrier-resistant and carrier-susceptible samples, as mentioned by Maria in result section. Almost all these proteins with significantly altered expressions were also detected in human dental pulp in their previous study in the year 2014. In addition, alpha-1 antitrypsin, phosphoglycerate kinase 1, peroxyredoxin 1, serotransferrin, topomycin alpha-1 chain, and vimentin were also observed in another study in the year 2005 on the human tooth pulp tissue. On this slide, I am going to talk about the biological function of some of the proteins found in this experiment, such as some carrier-resistant proteins such as alpha-1 antitrypsin and ATP synthetase subunit beta and phosphoglycerate kinase 1s are related to metabolism and energy production. Alpha-1 antitrypsin is a major circulating and tissue inhibitor of serine proteinase. It may also exhibit anti-inflammatory activities independent of protease inhibition action. Hence, increased level of alpha-1 antitrypsin could support inhibition of the activity of serine proteinases in tooth tissues, leading to the elevated resistance of teeth to the development of tooth caries. On the other hand, ATP synthetase subunit beta and phosphoglycerate kinase 1 are important enzymes that provide energy for the cell to use. ATP synthetase enzyme catalyzes the final coupling steps of oxidative phosphorylation to supply energy in the form of ATP, and phosphoglycerate kinase 1 catalyzes an important ATP generating steps in glycolysis. Although their connection in the prevalence of dental caries remains unclear, but it is interesting to know that both the above enzymes are upregulated in carrier-resistant patients, which could indicate an enhanced cell turnover and production of ATP in teeth. Apart from that, annexin A2, annexin A5, and vimentin are the structural proteins involved in cytoskeletal organizations. Annexins are soluble proteins that belong to a family of calcium-dependent phospholipid membrane-binding proteins and are involved in various cellular activities such as cell division and calcium signaling. Vimentin belongs to a large family of intermediate filamentous proteins that play important structural and functional roles in the formation and regulation of cytoskeleton. On the other hand, some caries-susceptible proteins such as pyridoxine 1 is an antioxidant enzyme that catalyzes oxidation reduction processes and is able to serve as a redox biomarker in various human diseases. Succinyl CoA 3 keto acid coenzyme A transferase 1 plays a crucial role in ketone body metabolism. Hence, these overexpressed proteins in carrier-resistant samples such as annexin A2 and annexin A5 and alpha-1 antitrypsin likely to involved in the natural resistance of teeth to the development of dental caries.
In conclusions, this paper made a deeper analysis of the experiment comparing a previous article published in 2014 by the same authors, improving their outcomes. It is important to mention that this is the very first paper to analyze proteomic 2D types in order to illustrate the changes in expression in the tooth pulp of caries-resistant and caries-susceptible people. They found overexpressed proteins in caries-resistant and caries-susceptible samples, suggesting that the resistance of caries development is a multifactorial mechanism. It is important to highlight that this study provided 17 missing proteins candidates for the chromosome-centric human proteome project, who had already identified 4,332 proteins. We find this paper really interesting and provide information that will be helpful for future research. Although this article couldn't declare those proteins that can prevent from developing dental caries, surely can be really useful for the industry dental care.
In regard to the ethical issues, the experiment was conducted in accordance with the Code of Ethics of World Medical Association for experiments involving humans. Moreover, all teeth collected to perform the experiment were extracted in a dental clinic after acquiring the patient informed consent regarding the donation of tooth for research. In our peer review, we believe that this work should have been published, as it is the first time that someone presented a proteomic 2D Daj analysis to illustrate and compare the changes in the expression of proteins found in the dental pulp of caries-resistant and caries-susceptible people. Additionally, if anti-caries function of the proteins identified in the dental pulp of caries-resistant people is confirmed, then these proteins could become a part of commercial toothpaste, which will improve the oral standards in future. Although the article has not been cited yet, but it could be due to the reason that it has made available online just 4 months ago. Therefore, in our point of view, such kind of work will surely lay the foundation for the advancements in the field of proteomics.